Thank you, Dale. You're very kind. Probably more gray hair than wisdom, that's for sure. Yeah. So all of you hardy folks here this morning, great to see you. My wife texted me and said she heard that yesterday Saskatoon was the coldest place on the planet. Did anybody else hear that? Is she exaggerating, or it could be right? Anyway, Dale mentioned I'm at the university and I'm a plant breeder, and it's sort of maybe a funny combination because if I talk to my colleagues in Europe, it's kind of a different situation. In Europe, plant breeders work for companies, and university professors study the way genes work, the way microbes work. It's very basic research, and the breeding is over here. We have kind of a unique situation, I think, in Crop Development Center, not to brag, but where we kind of combine those disciplines in one place, and I think that we see some synergies that way. So we're doing practical breeding at the same time. We're also doing basic biology work, and we hope that the two kind of speak to each other and benefit each other. And maybe I'm going to ask one of the recent graduates of University of Saskatchewan. I see Shannon is back there, or maybe Corey wants to interpret this slide that you see on the screen. Maybe Dale could. He's a very wise man as well. So please, give me your interpretation of what you see here. And well, anyway, it looks rather busy. And this is a slide I borrowed from our colleague Judith Burston in France. And I find that when the Europeans put together their slides, they like to put a lot of stuff on there. Wow. And it becomes so small, I can't read it. I have these simplistic slides that you're going to see coming soon, where there's just like 5 words on the whole slide. But at least then you can read it from way at the back while you're drinking coffee. But here you see this is very dense with information. But why I put this up here is just to kind of give you a little of this impression about basic science together with breeding. So this is arising from a project funded, the Canadian component of it was funded by Sask Pulse Growers, and me and a couple of colleagues are part of an international group that developed the genome sequence for pea. And it's a rather big project. It spanned over the last 5-6 years, because these are kind of complex things. Maybe 20 years ago we had the Human Genome Project. It cost a few billions of dollars to do that thing, involving hundreds of scientists. So over the years, that kind of work has gotten cheaper and more efficient, but nevertheless, it's still a massive project. If some of you have heard Curtis Posniak, my colleague, speaking in some kind of meetings, he's been heavily involved in the wheat genome sequencing efforts. And so likewise, myself and Bunyamin Turan, who's a chickpea breeder, and Kishore Gali, who works with us, have been involved with this project on pea. And so this is part of a publication that's now going under review in a rather high-profile journal, I would say, and hopefully it will be published there pretty soon. So anyway, thanks to you as growers, actually, for supporting that work. And you might think, why do you want to support this kind of work? But it's sort of an investment into the medium term, the longer term, because this kind of info becomes really a resource for us when we want to look at breeding for traits of interest. If we know the regions of the genome where these traits are located, then we can start to develop markers related to those things and start to do a better job of breeding. So here's another kind of a picture that we as breeder geneticist type people like to put on the screen. These little cartoons are depictions of pea chromosomes. And so this is coming from a paper that we recently published. Basically, it's a way of us trying to map out the regions of the chromosomes that are related to things like lodging resistance or time to maturity or the iron content in the seeds. So these are the kind of things that You're going to hear more and more from us, or you already have been hearing about them, but that's going to continue. A couple of other examples of some recent research that I've been involved in. This is work really led by Rosalind Buchert, who is a plant physiologist in our department. These are from two separate projects of her grad students that are just recently completed their PhDs. The overall theme of these studies is what happens to the pea crop when it's hot outside. If you remember this spring, we had some hot days, we had some hot weeks, and it's not just pea, but many of the crops that you grow are going to be impacted by these temperatures that are, you know, upper 20s into the 30s are going to start causing some issues. So in the one case, you see pictures with these flowers and pods, and you see what's the effect of the heat on that. On this side, it's more so related to what happens to the canopy. So for a little example, as the temperature gets warmer, the duration of flowering gets less. And we don't really like that. It's always better if the crop is flowering longer in terms of getting good yield, but that's what we know. Another kind of different type of work that we've been involved in recent years, this is led by Tony Yang. You see some other names here, Rosalind Buchert, Jeff Shano. Many of you know Dr. Shano from soil science. Axel Diedrichsen is at the Gene Bank, and Hossein Zakari is actually a former grad student of Rosalind. So in this study, Tony looked at the effects of different rhizobial strains on 3 different kind of pea types, and you see there's kind of 2 things happening in these charts. I think I can't read it from here, but I think this is the above ground biomass of the peas, and you see that this type compared to this type to this type. So the pea themselves differed in how much biomass they produced, but depending on which rhizobial strain was applied, there were also differences. So we saw some interesting interactions, and that's something that we want to follow up with this work going forward. Another type of work that we get involved in on the campus. Oh, thanks. Now I can see the screen right here. Excellent. This is work led by Mike Nickerson. His master's student Angie Lamb did this work. Many of you, I don't know, does anybody know Mike Nickerson? Okay, you know Mike. Okay, of course you know Mike. So if you know Mike, I'm telling SPG and Ministry of Ag, you guys should consider inviting Mike to speak at one of these meetings. He's a very entertaining guy. He knows all about food science. And Bob Tyler, maybe, does anybody know Bob Tyler? Okay, many of you know Bob because he's, if you can invite him to speak here too, he would be excellent. But anyway, for example, this particular study, Angie developed pea protein isolates and compared them to other protein types that were on the market. So in this case, 2 different soy ones, whey from milk, a commercial pea, a wheat protein isolate, and egg. And then looked at how do they compare in terms of solubility, oil holding capacity, foaming capacity, and so on. So some of the different traits that are important in the food industry were evaluated here. So I think what I'm trying to say is that being on a university campus, we have many linkages with other researchers, and it makes it kind of a dynamic setting. It makes it an interesting setting, and I think it can benefit us as plant breeders. So, I mean, I'm interacting with other pulse breeders in our own group. Many of you know Bert Vandenberg, who's breeding lentil and faba. Bunyamin Turan is breeding chickpea. Kirsten Bed is breeding common bean, and Sabina Benissa is a pathologist working on these crops. So we have our own kind of internal linkages within the department. I mentioned Rosalind. Billigatu is the forage breeder, so we're doing a bit of work with forage pea. Within the college, Nickerson, Yongfeng Ai. Yongfeng is a young professor. His expertise is in starch and other carbohydrates, so I'm involved with some work with him. Bob Tyler I mentioned. Jeff Shano, Diane Knight in soil science, Greg Penner in this forage work that we're doing. He's a beef cow nutritionist, so looking at what happens when we feed bales made from a mixture of pea and barley to beef cows. Richard Gray is an economist. Maybe many of you know Richard. He's a senior ag economist, and in certain projects we're looking at not just the biological benefits, but for example, in a soybean Genome Canada project that Richard and I are both involved in, we're looking at what happens economically. What are the potential benefits or pitfalls to introducing soybean to Western Canada as a crop? Ag Canada, so we interact a lot with Ag Canada researchers. DJ Bing is a pea breeder at Lacombe. Yantai Gan is an agronomist at Swift, who's done a lot of work on pulses over the years, and maybe you've heard him speak. Jim House at University of Manitoba, we're beginning to work with Jim quite a lot. He's an expert in protein quality, and so that's an area that we're expanding our work on. Francois Belzile is a genomics expert at University of Laval, and I've mentioned the pea genome sequencing activities. So this is kind of leading me to another kind of thing that we as researchers like to get involved in, because we like to meet our friends and colleagues in different places to share our results and think of new ways to address problems or topics. So in May, I'm going to go to Dijon for the International Conference on Legume Genetics and Genomics. This conference is held once every 2 years. Here's the advisory board of that, and you'll notice Kirsten Bett, my colleague, is on the advisory board of that conference. And it just so happens that the following week, there's another conference on legumes, which I am planning to attend in Poznan, Poland. The previous one that I mentioned here is really focused on genetics, genomics. This one is focused on let's say a broader set of topics, so agronomy, breeding, end-use quality, economics of all kind of legumes, not just pulses, but even forage legumes or tree legumes. So this one's in Poland, and you can see that I'm on the scientific committee of that. So these are ways that we can kind of enrich ourselves, I think, as plant breeders. So I'm going to switch my gears a little bit and talk more about plots and trials. This is a long list of locations where we've grown the regional variety trials of pulses. In this case in 2018, but we've used similar locations in earlier years. And I'll just give you a few snapshots of some of those trials. At many of these locations, we also have some of our breeding nurseries. But the regional trial is the trial that gives rise to the data that you see in that yellow booklet right there. I can see one on the table. This updated every year, the Varieties of Grain Crops booklet. So I'll just, a quick little walk through some of the plots from 2018. Our most southern location is at Limerick. It's near Assiniboia, and it tends to be a bit of a drier area. In this case, we're on the farm of Barry Reisner, who's a seed grower that many of you may know. Over the years, that area is known to have pea leaf weevil, and you're going to hear more about that from James after lunch, and he can tell you very nice stories about all of these little insects that we should pay attention to. But I was happy to see that we didn't really see much of leaf notching this year at that site, so that was kind of positive. Rostern, a little bit north of Saskatoon, we're with a long-term cooperator on farmland between Rostern and Duck Lake. It's usually one of our high-yielding locations. Here we're in the city. We have a bit of land right in the city along Preston Avenue. We're not sure how long we're going to be able to hang on to that patch of ground. We lost one chunk of it recently due to the new hockey rink, and there's rumblings that other parts of that might get lost due to student housing or something. But for now, we're kind of keen to retain at least some land within the city. And actually, we've moved our main within city work to the area just a little bit north of College Drive. It used to be operated by Animal Science, so our irrigation system has moved there. So here we have some faba and pea. Kamsack would be our more easterly site, nearly at Manitoba. We're on the farm of the Fedoric group at Campsack, and just taking note of some pretty nice nodules there. Back in the city, I'm doing a bit of work on soy. My main effort is on pea, but we have a small program on soy. I'll tell you a little bit more about that. These are some little plots that are, we grow a whole bunch of them. We don't harvest them all. I go through them as they're maturing, and I kind of just select the ones that look best, and we combine those and carry on. And that's kind of the approach that we use in all of the pulses. These are F3 generations, so it's near the beginning of the breeding program when we have high numbers, and as you know, plant breeding is a process of narrowing down from many to few. So in pea, for example, I might have about 10,000 microplots, F3 microplots, and eventually that's going to give rise to 1 or 2 or 3 varieties, I hope. Here's what we call the SPG farm. It's land owned by Sask Pulse Growers. It's near Floral, Saskatchewan, not just a short drive from the city. And we've used that site over the years for pea, faba bean, lentil, basically all of the pulses. And it's a good site. The land is a little bit sandier than Kernan, which I'll mention in a moment, which is right near the city. There's a little bit lighter land, and so it gives us another idea of things. I want to just point out a little bit about how we make breeder seed. So all of these plots, you see they look pretty uniform to each other, and that's what I was hoping for in this case, because they're all derived from one variety. So it's sort of a process of variety purification, these long plots. Each plot traces back to a single plant 2 generations earlier. So we grow these out, we look at them in the field, and then we look at the harvested seed after, and anything that's unusual from the mean, even if it's better than the mean, we throw it out, because we want the mean, the average, to look uniform, because we bulk it all, right? So that's the process that we use. The cereal breeders use a similar approach. I mentioned forages a moment ago, and this is just a multiplication of CDC Horizon, which is a forage type pea at the Livestock Center near Clavet. Lucky Lake, we're on the farm of Sean Buer, former chair of Sask Pulse Growers. Again, it tends to be maybe a little bit on the drier side compared to the more northern sites, or the more eastern sites, but we've had nice results there, and here you see some nice chickpea plots on Sean's land, and here are some good lentil trials as well. Kernan, I mentioned, is just on the edge of the city. In fact, the city is stretching and encroaching and reaching up and basically bordering Kernan. It makes it a bit tricky. We don't fly airplanes spraying insecticide over Kernan. We used to. We don't anymore. Too close to the city. But it's a place where we multiply breeder seed, and this is what it looks like. And here are a few of the recent pea varieties. Well, I'll just mention quickly. This is CDC Lawachko, one of the recent yellow pea. Next one is Forest green pea, and this is Dakota, which is not new, but there was some new demand for breeder seed of CDC Dakota. It's a dun type, so it's a type with purple flowers and pigmented seed coat. Rostern, I mentioned, tends to be one of our high-yielding locations. We grow, you can see here, there's pea, faba, lentil, and pea again, different nurseries for those crops. It's also quite convenient because only an hour's drive from the city, but that's proven to be a very good site for us. A small amount of our research activities takes place under irrigation. Maybe 10–15% of our total work. The rest is on dry land. So we make use of we collaborate with people like Gary Natowich at Outlook, and in this case we're at Broderick, just a little bit south of Outlook, and here we have some soybean, we have faba, and common bean at this location. One thing we noticed, well, you folks, I'm not sure how intense you experienced drought this year. My impression was most of the area of Saskatchewan south of Saskatoon had some drought, and no doubt here at Limerick it was on the dry side because peas were maturing already at the end of July. I'd rather they were maturing toward the end of August, because that would typically, you would think, give rise to more yield. You can see there's lentil plots in the background, and they're also turning quite a lot by July 24th. In fact, even chickpeas were maturing at that location. And historically, maturity has been a big challenge in chickpea, but in a dry year, away they go. Swift Current, here's a lentil regional trial. You see nice plots here and some differences in their time to maturity. Chickpea, maybe you can see a little bit of crop turning on July 25th, but probably a week later you would have seen a lot more. Camsack on August 1. Here's some faba doing quite well on August 1. Dry beans from Kirsten's program. She's breeding all different types. Pinto, black bean, yellow bean are the types that she's putting the most emphasis on these days. And here's soybean at Camsack. It was kind of late by the time I got to Brandon that day, so I ran as fast as I could to take a few notes before it was lights out. And we work with folks at Indian Head, just down the road from here. And it's been a good site. It's a good cooperator site for us at Indian Head, especially on pea. Now we're into August, and you see the peas are maturing. Soybeans still green. A lot happens in soybeans in August, in my experience. You could be at August 1st and you see very little tiny pods, but then boom, they start filling up as the month of August progresses. We work with our colleagues in Alberta, especially Alberta Ag. Mark Olson and Christy Hoy are running trials of pea, faba, together with us and with Ag Canada folks, and even with Lima Grain folks. They kind of cobble together some funding, and they grow some really nice trials. So they have trials at Vegreville, Nemea, which is just north of Edmonton, and then up at Barrhead. So I like to have a look at those. Here you see that in contrast maybe to Limerick or Lucky Lake, at Barrhead they had plenty of moisture, kind of lush growth, lodging, later maturity, but they got them harvested, but I think it was into mid-September before they got those cleaned up. Lacombe is usually also a site where it's usually a bit cooler, high-yielding location, but this year it was a bit hotter there. Chickpeas are mature here. Peas are mature here at August 20th at Melfort. Soybeans are maybe starting to turn a bit. Common beans are turning nicely at Melfort, August 20th. And here we're starting to see maturity August 28th. So, if I was giving this talk 5-6 years ago and I was showing you soybeans maturing in August, I think you might have been surprised. I think I would have surprised myself, but it's a function of the germplasms getting earlier and maybe a little bit of the seasonal effect. However, we did get a touch of frost on September 5th at Saskatoon, so this is one plot that was really a late, I think it was accidentally put into this nursery, a type that was more adapted to Ontario, but it was fun to watch that one because it really was very green all summer long, and then got a little bit nipped by even a -1 on September 5th, whereas the rest of the nursery in Saskatoon was fully mature by then, as it was at Roster. Dale named this guy Chocolate. I don't know, I guess there's a reason why he named him that. But, we had strange visitors sometimes. Sometimes I'm not surprised to see a rabbit or a gopher or two, but okay, this is in the middle of Saskatoon, so here we go. What are we trying to do in pulse breeding? I mean, the general comment in whichever pulse crop it is, pea, lentil, chickpea, is we want to improve the crop for the field, for how it performs for growers, but we also want to improve it for how it's going to be accepted and utilized and benefited from in the marketplace. So that's what we're trying to do. Pea and lentil are grown a lot in Canada. You folks are doing a great job of growing those crops. Here's a little snapshot about varieties, in this case of yellow pea, insured acres, data from crop insurance. So CDC Meadow, CDC Amarillo are the two that are grown the most, Saffron third. And then there's a bunch of others that are grown a lesser amount. And here I've taken these varieties and moved them onto this table, and it's just an excerpt out of the seed guide. Just want to point out a couple of things. We're using CDC Amarillo as the yield check at 100%. We're reporting the protein concentration of pea nowadays. This is the second year we've added that column into the guide, because we have companies that are fractionating peas now, or starting to, and they're asking, well, which varieties have more protein, which have less. So we're reporting that now. 23 is the average for Amarillo, and then the others are just indicated plus and minus of that. I want to feature a little bit CDC Inca right here, because there's pedigreed seed, certified seed is available. This is the first year that we have large amounts of certified seed of Inca. You saw up here that Meadow is the most widely grown. Inca's down here about 6th or 7th place. I feel like Inca should move towards first place, because if you look at the average yield, Meadow 91, Inca 102. So you can get a 10–11% yield advantage by switching from Meadow to Inca. Not much reason not to do that. So if you're due to change up your varieties, that's my suggestion for this year. One thing I forgot to mention, so with Inca, good seed coat breakage score. Unfortunately for Amarillo, only fair. So you're going to have a strong seed coat with Inca, similar to that of Meadow. And a couple of new ones to keep an eye on, Spectrum and Lawachco. I'll talk to you about those maybe next year. So far, about 90% of the insured acres of peas, if we take yellow, green, and all of the pea types together, are coming from the CDC program. Thanks to you folks, thanks to the Variety Release Program for boosting that along. Probably that number is going to go down a bit in the years ahead. I wouldn't doubt it, because there's good material coming from Ag Canada, from Lima Grain, from DL Seeds. But hopefully we can still do a good job at the CDC and keep a good proportion of the market. Here's the greens. They're growing at a much lower scale than the yellows. You see several choices. I want to point out again on this protein theme, there's a variety called Limerick that we didn't breed it for high protein, but it kind of came along fortuitously. Sometimes that happens in plant breeding, and it's almost 3% higher in protein than CDC Amarillo. So in our newest project, we want to kind of shift some of that higher protein genetics into the yellow pea background, because it's yellow pea types that are favored by the fractionation industry. Keep an eye out, especially for CDC Forest. You can get 20% more yield than Striker or Razor if you go to Forest. That's quite a big boost. I think it's worth changing varieties. Chickpea. Chickpea, in contrast to pea and lentil, where Canada is a very big player in pea and lentil, chickpea we're more of a minor player. But it's important nonetheless, and it's an interesting crop for export. And you folks have become very good at growing chickpea. But chickpea, really the world leader in chickpea is India by far. For 6 years I was also breeding chickpea when I first came to Saskatoon. And I went to a conference in the middle of India, a chickpea breeders meeting, and there's like 40 breeders, something like that, of chickpea just within India. 40. In Canada we have 1. Anyway, that shows something. There's also a lot of people in India, but 40 chickpea breeders. You folks are good, high-yielding producers. Highest yielding chickpea producers on average in the world, so you're doing a good job. New varieties, Leader and Orion, have gained popularity. little bit bigger seeds than Frontier, which was grown more so in the previous few years. You can find this info in the seed guide, the variety, the type, Kabuli, Desi, percent yield of Amit, the Czech, Ascochyta blight, the lower number is better, seed weight, typically you get a premium for large Kabuli, as you know, and a new piece of info is whether or not it's tolerant to the IMI herbicide, and you can see 2 of them are. So take note of those features. And Bunyamin asked me to point out that the new varieties coming will be with that herbicide resistance, that group 2 resistant trait. Here's one Kabuli that's released to seed growers this year, and several of these could be coming in a year or 2 from now. They're being multiplied in Arizona right now. We use a winter nursery in Arizona for pea and chickpea, and in special cases that can really help us to move things along a year faster than without that. As plant breeders, we can't succeed unless we have variability. If every variety looks the same, if everything is genetically the same, we can cross them, but we're not going to make any progress. So we like to cross more diverse material. Maybe in some ways we do that by, okay, I'll pick a pea variety from here and one from Australia, cross it, and develop some new things. But in chickpea, at a certain point, there's not that much variability, or there is some, but they're looking for more. So to go a little bit more extreme, they're even going to some wild relatives. So you see even how the seeds look. Here's a typical Kabuli. Here's Cicer reticulatum or Cicer echinuspermum. They look very different. And you think, wow, why do you want to use something like that? Who's going to buy something like this or this? Probably they won't. Probably most markets won't buy that type. But if you cross this with this and then kind of select this type back, a lot of work. It's not something that you can do in a 1-year project, but in a 5- and 10-year project, you can start maybe to bring in some of the interesting traits from reticulatum, which could be more drought tolerance, more iron concentration in the seeds, higher yield in some way or other. So we like to have variability. Very briefly with soybean, the 2 main goals are earliness and yield. Those are the 2 main goals for our small program on soy. Currently I'm involved in a small breeding project. It started 5 or 7 years ago, working especially with Elroy Kober, who's the breeder at Ag Canada in Ottawa. The program in Ottawa has been running for 30, 40 years, so it's very established. If you remember, if you know about soybean, there's like Maple Arrow, Maple Amber, all that Maple series that came out of the program at Ottawa. And they've had an emphasis on short season soybean for a long time. I should point out that Elroy is kind of a world expert really on earliness, early maturity, early flowering, the genes that are involved in that phenotype in soybean. He's a world expert on that, so he's a good guy to work with. Louise is a breeder and a molecular biologist working for the Sarome cooperative based near Montreal. So all 3 of us are interested in short season soybean, be it for the short season regions of Ontario, Quebec, the Maritimes, and in my case, more so for Western Canada. So we're working together on this. We hope we can make some progress. This is a little bit of info arising out of our previous 5-6 years of work. We started by, I would make selections out of Elroy's populations in Saskatoon, selecting under our conditions in material that's still variable, and then the good ones, we just carried them forward year after year, tested them for yield. So this is a little bit of data from 2018 from 5 locations: yield, days to maturity, protein, oil. And if you look at these 2 in the bottom, these are the breeding lines, these 4 are the checks. You see some numbers, 107, 111 in yield, not bad. Days to maturity, 109, 113, so it's in the range of the checks. Protein and oil are within the range of the checks. So we may have something that we can offer to you folks pretty soon. Root rot, we've heard a little bit already today. from Sherry Lynn about root rots. We heard quite a bit from her. I'm just going to add one little note to that in terms of breeding for improved root rot resistance. We're fortunate to benefit from some of our international colleagues from France and from Pacific Northwest of US who have been working on this problem for a long time. They've had more trouble with Phanomyces than we have, or they had problems with it earlier. So they worked hard on this and have even developed some DNA markers linked to some resistances in pea. And so, if you just generally look at this, here's CDC meadow. This is root discoloration when exposed to a good shot of a Phanomyces, and you see it scoring like 4, where 5 would be the max. Here are 2 breeding lines from our program, kind of similar to meadow, but here are 2 different sources of partial resistance, and so they have a quite much lower score. So basically what we're trying to do is to bring in resistances from these 2 into material like these that are going to grow well under your conditions. That's our goal, and we've really taken kind of a molecular breeding approach. It's not typically what I do, but in this case we have access to these resources. So it involves basically crossing the resistance source here with a CDC line, this other resistance source with the CDC line, and then using markers to select the offspring that carry the genes for resistance, and then intercrossing source 1 and 2 so that we can kind of stack them together. And gradually we're going to bring those to the field, and hopefully all that work will give rise to something that looks better in the field. In the case of lentil, similar idea, but starting in a slightly different place. The cultivated lentil didn't really have much for resistance to Phanomyces. So Sabina started investigating Lens orientalis, and all of these lines are different Lens orientalis. These are relatives of the cultivated lentil. On the very far side is CDC Proclaim. In this case, everything is indexed onto Proclaim, so it's not that Proclaim is any worse than any other lentil variety. I don't want to give that impression, but she just indexed it to 100, and some of these are 50 or 20, or a few of them even look much better. So she has something to work with, and that's what's going to be the focus of a new project funded by Ministry of Agriculture. Thank you. Sherry Lynn also mentioned that not only do we have a Phanomyces, but in many cases there are various fusarium diseases that are also root rotting problems. And so we're looking at that together with Shamit Chatterton, who's a plant pathologist at Lethbridge, Ag Canada. She's doing great stuff on root rots. Here are the pea varieties that are in the regional trial this year, screened by Sabina for again, sort of discoloration caused by Fusarium avinaceum, and you see there's a range of responses. The best ones are these 4, and these 4 are all purple flower types with pigmented seed coat. And we've known for some time that some of those phenolic compounds in the pigments in those seed coats are going to exude into the root zone, and they're a bit antifungal. So it's not surprising that those are the best. But you don't necessarily all want to grow purple flowered peas. But I'm still happy to note that at least some of the white flower ones were at 30 instead of at 70. So we have something to work with. Similarly, in lentil, we have something to work with. These are the lentil varieties in the regional trial. You see a range of responses. It's lunchtime. I'm smelling nice food. And now I'm going to show a few pictures about food. So, I wonder, when we load up our plates, are you folks going to put veggies on one half, whole grains in this bottom corner, and some proteins in the upper corner? Are you all going to do that? I'm watching. I'm going to go do a little survey around your tables and see about that. So, this is the Canada Food Guide, the new revised Canada Food Guide. I read that in France, they revised their food guide just recently, and when I looked at what they did, this looks a lot like this. So you can decide if you really like this new food guide or not. I've had mixed responses when I showed this picture at the previous 3 locations. The Western producer gave it kind of a cool response as far as I could tell. You know, not all proteins are not created equal, and that's very true. Talk to Jim House, my colleague who's the expert on protein quality, and he'll definitely say that. But one thing that, you know, we should keep in mind is that proteins can be complementary, and this has been known sort of since time immemorial, as they say. When you look at where crops were domesticated in the world, often in the Mediterranean region, in Asia, or even in the Americas, typically what you see is that a cereal and a pulse are domesticated in the same region. In the Americas, we had corn and beans. Other places you had like lentil and wheat. So when you consume the two together, boom, you have essentially a full protein. So there you go. Seems to me there's quite a few pulses in that corner, so this is a pulse meeting. I guess everyone's really happy about this food guide. A recent, quite a big paper, it had a little bit of fame or notoriety, depending how you look at it. It was published in The Lancet. This is one of the top medical journals in the world, and they talked about, you know, what kind of food habits should we as humans have with respect to our health and with respect to the health of the planet. And so they basically recommended some things that are, you know, kind of in line with the Canada Food Guide. You can read it if you like. Trends. This is a slide from Pulse Canada I borrowed, and there's been, from 2014 to 18, among the top 10 food trends, protein, plant-based foods. Okay, so these kind of, it's a buzz. It's coming along. You see things like this. Beyond Meat burger. After water, the first ingredient is pea protein. It's good for your industry. Here's a little comparison. Beyond Meat has pea protein and beet juice. Impossible Burger doesn't have pulses, but has wheat and potato protein. I haven't eaten this one. I've eaten this one several times. This one has heme. Interesting. This is a little bit of a legume link, but basically heme is coming from leg hemoglobin out of the nodules of soybean. And again, it's sort of to give red color. So there's various new products, beverages, and so I think these kind of things give more demand for pulses, and you know, should help, I think, in the long run, improve your market opportunities, prices. This one is showing protein powders, and pea is being used quite a lot in protein powders. How many minutes, Dale? 8. Okay, thank you. I'm just about done. So you've read in Western Producer and various places about expansion in protein extraction business in the Canadian prairies. It's not just in the Canadian prairies. Some of this work, Roquette, for example, their main office is in France. Cosucra is extracting protein there in Belgium. I think AGT has already been active in North Dakota for some time. So Parheim Foods here, not here, I think I'm not in Saskatoon. Parheim Foods in Saskatoon has been extracting protein, starch, fiber out of peas for some time. So there's some momentum has built up, and the momentum is going to feed into various food industries, into pet food industries, into aquaculture industries across the prairies. I'd like to acknowledge Sask Ministry of Agriculture for supporting these 4 projects recently involving work that we're doing at the university on pea. The first one really is focused on this protein theme. We're trying to boost the protein concentration in peas, also the quality. When I say quality, it's, we talk about the amino acid balance, sort of this thing about complementary proteins. We know that pulses in general tend to be a bit low in so-called sulfur amino acids, methionine and cysteine. If we could find types that had a bit more of those, of methionine and cysteine, that would be beneficial. It would increase the so-called PD-Cas score. It's sort of an international score that we use to talk about the quality of a protein. The higher the number is, closer to 1, the better. Right now, pea might be at 0.65 or something. We'd like to boost it. Can we get it closer to 1? So if we can find types that have more methionine and cysteine, that would be good, but there'd be a few other amino acids that if we could boost them a little bit more, that would be beneficial. So that's part of what that study's about, really studying it at the genetic level, at the chemistry level. We have a separate project where you might have heard me talk about biofortification in various talks over the years. So here we're really looking at iron. Can we boost iron in peas? Especially iron that's available when someone eats it. We know that it's not enough that we increase the concentration of iron, but it has to be available to the human. So we've done work on this for the last decade or more, some of it funded by Sask Pulse Growers and the Ministry of Agriculture. So now we're at the point where we're going to say, well, okay, we know that a certain type of biofortified pea that we've developed does deliver more iron into human cells when we've done the work in a Petri dish analysis. We want to take it 2 steps further. One, we're going to feed it to chickens, and that experiment is going to be happening in the next month or 2 with our colleagues in New York State. They're going to feed biofortified pea to chickens versus a control pea and see what happens. Do they grow as well? What's their hemoglobin status? They'll check the blood at the beginning, at the middle, and the end of the study. I think it's a 6-week study, I believe. And taking it a notch further is going into humans. So we're going to have a study starting this year where we're going to be feeding these kind of 2 different diets to athletes at University of Saskatchewan. And hopefully they are able to enjoy the diet for 3 months straight. And then we'll see if we have to, I'm open to suggestions here, but we have an idea like how we're going to feed this thing. And so we're going to make kind of like a granola bar product, like a sports bar type of thing. We hope if we make it taste nice enough, they'll be willing to eat it for 90 days in a row without skipping. Let's see what happens. I hope they can go for it. But it should be quite interesting, I feel. Glycemic index is another kind of nutritional thing. We're talking all nutritional things, and I'm holding you away from your lunch. But glycemic index is simply a thing where it's how fast do your carbohydrates switch from complex to sugars. So if you eat a piece of white bread, it'll break down fairly quickly to simple sugars, and it has a glycemic index of 100. If you eat lentil or pea in a bowl of soup, maybe the glycemic index is 50 or 60, so it breaks down a bit more slowly. It's a more like slow release energy, which is good, especially if you're diabetic, but even if you're not diabetic, kind of having a slower release is probably better for your health. You sustain your energy longer, you don't get spikes in sugar and stuff like that. So what we thought was, okay, if peas on average are 60 in glycemic index, well, are they all 60, or some of them are 70, or some of them are 40? Maybe a peas that are at 40, in a way, from a nutritional point of view, might be better, and we could brag about that and promote peas in that way. And the final one is back to the topic of nitrogen fixation. I mentioned the study from Tony Yang where he shows some differences in how certain pea varieties respond to different rhizobial strains. Can we move forward into breeding with some of that material to select types that actually do fix more N? I think that would be quite nice to see. So we work with a big group of people. I mentioned there's 4 breeders in our group, and we have about 30 staff that work with us, that work on all of these crops, and they're growing the trials and taking care of them, harvesting them, weighing them, getting the seeds set up for the next year. So as soon as we harvest our plots, it's amazing. We might be harvesting in September. By October or November, definitely they're already setting up the trials for next year. So they just switch it from one into the other. So they do a lot of work. I've mentioned about breeder seed and releasing varieties, and so these guys are the ones who are developing the breeder seed. They multiply it from, we might supply 100 kgs of a new pea variety, and they'll boost it up to 2 tons or so, and then that seed will go out to growers. And so thank you for listening. Acknowledgements to funding organizations, including SPG, Ministry of Ag, WGRF, CCAN, and others. And thank you. Lunchtime.